changed in chapter 2 is this one. So what I did, here's from chapter 2. This is just in general symport and antiport. And the confusion with symport and antiport is always, is it active or is it passive? Because it could be either. It's not easy to classify. It depends on what ions are moving. So here I give you a rundown of the transporters that I will probably be talking about in an exam, the ones I really know. Sodium glucose and also sodium amino acid transporter. These are going to be active because sodium is being used as the driving force for bringing those into the cell. Sodium calcium exchanger is also active. Sodium is being the driving force here. Now here you've got sodium and hydrogen. Both of those could be power sources, so it's facilitated diffusion. It's neither of them is really going to be actively moved. And then our chloride bicarbonate transporter also is antiport and facilitated diffusion. We'll see this perhaps today if we get as far as the spectrin cytoskeleton. That's called band 3 when we get there. So we'll talk about that some more. So here's basically your list of active versus passive transporters. Okay, so now let's go to chapter 3. Am I showing chapter 3? No. Hang on, share chapter 3. Let's go big or go home, right? Okay, quick review of quick review of last Wednesday's lecture. So these proteins are called ARPs, actin-related proteins, and these are what are going to be actually doing the polymerizing, depolymerizing of the microfilaments. So these 4 are the polymerizers. These 2 are the depolymerizers. So formin is the enzyme that's actually adding the monomers. Profilin brings the monomers over to it, and thymosin beta 4 is kind of holding on to those monomers when you're not actively polymerizing. The thought there being actin will spontaneously polymerize at the concentration it's at here in the cell. So thymine kind of holds on to that so it doesn't keep polymerizing. And cap Z, once you've stopped polymerizing, you need to cap that plus end, because remember, your actin monomers have ATP attached, and they break that down to ADP. And when they do, the microfilament is destabilized, and it can fall apart if it's not stabilized. So your cap Z is stabilizing the plus end of that. And then for breaking things down, ADF cofilin is at the minus end, chewing out individual monomers, and gelsolid cuts in the middle, so then it produces more minus ends that ADF cofilin can use. Okay, and then you can watch this animation later if you want. I think it's really cool. The other thing that's important is branching, and branching happens mainly when you're talking about cell crawling and the lamellipodium. And so in order to branch the fiber, basically what happens is CDC42, which is a small monomer G protein, will be activated by some receptor, and then the receptor, and then CDC42 will activate WASP, and WASP will grab ARP23 and put it on the side of an already polymerized microfilament and start a nucleation point so you can branch off into that direction. So this is a little animation you can look at later if you want. This is kind of showing how that cap Z is regulated. So when your chemoattractant, whatever it is, it could be a growth factor or something, activates the receptor then activates this enzyme PIK. PIK is phosphatidylinositol kinase. So here's PI, and it's going to turn PI, which is this guy over here, into PIP2. It's going to put a phosphate here and here. And that is going to attract cap Z, so it comes off the plus end and opens up those plus ends for polymerization. Another thing that's going on with our PI is you've got phospholipase C here, which again, we talked about chapter 2 a little bit. Phospholipase C will cut that PI here so that PIP2, that whole inositol trisphosphate, will come off. When that happens, then that releases these guys. And these are the ARPs we just talked about: gelsolin, cutting in the strand; cofillin, depolymerizing from one end; and rofillin, which is the transferring the monomers over. So it's, as you're breaking down this PIP2, you're releasing those, and then they can get to work on the microfilaments. So basically, you're turning over PIP2. You're making more of it, but you're also breaking down what's there, and that stirs the pot basically, and all the ARPs get activated. Oops. Okay, and why are we doing this? Chemoattraction, which is cell crawling. So basically, you got a source of some chemical, and the cell is moving in the direction of that force. So the example I like to use is you've got some kind of damage in a tissue, and blood spills out, platelets get activated, and then platelets release PDGF, platelet-derived growth factor, and then that draws cells like fibroblasts into the area to start putting down collagen and fixing the damage. So that's cell crawling. Now, there's several structures in the cell that are important for cell crawling. One are the filopodia, which are long projections sticking out of the cell. And what's important to know about these is they are parallel arrays of microfilaments, and very similar in structure to the microvilli in the small intestine. So that's this. These microfilaments are being cross-linked by vilin. Let's see, just vilin and then fibrin, and they're attached to the plasma membrane by myosin and calmodulin. So those are your parallel arrays. And then you've got anti-parallel arrays, which are going to be the stress fibers. And these are anti-parallel, just like two thin filaments in a skeletal muscle are anti-parallel. We'll see that today. And they're that way because you can get a myosin sitting between them, grabbing one, grabbing the other, and pulling them towards each other. And that's how the cell kind of moves itself into that lamellipodium, by dragging itself forward using this process. The next thing necessary for cell crawling is your lamellipodium, which has to do with, this lamellipodium reaches out in order to start crawling in that direction. And you've got branched microfilaments in there. Now, another thing I just skipped over real quick are the cross-linked strands in the cortex of the cell. So these are cross-linked by this protein filamin, which is a dimer. It binds to two different microfilaments, and that creates a connection between those microfilaments. And that makes up the cell cortex, which surrounds the outside of the cell and supports the plasma membrane, holds it in place there. And then here, getting back to our G proteins, this is all the polymerization of all these structures. Typically, in a cell that's not crawling, they're not there. Once the cell gets stimulated to start crawling, it's going to activate these G proteins, RAC-Rho CDC42, and they're called Rho GTPases. They're all very similar in structure. So Rho is producing stress fibers, RAC produces lamellipodia, and CDC42, in addition to branching, is producing filopodia. So basically, when a cell gets activated, all of these G proteins get activated, and the cell just rearranges its cytoskeleton to start crawling. And here is the cell crawling itself. And 4 steps: protrusion, attachment, translocation, and release. And they're all happening at the same time, basically. But first, your lamellipodium extends out, that's protrusion. And the interesting thing about that is it's going to treadmill here for a little bit. It's kind of like figuring out what it's up to. So your myosin is grabbing stress fibers and pulling them back, or microfilaments at any rate, pulling them backwards, and they're polymerizing up here and depolymerizing here. So it's just kind of like a treadmilling going on until attachments get made between this microfilament and the substrate. Usually it's extracellular matrix proteins in vivo. And once that microfilament is attached, now when this myosin pulls on it, it's not pulling the fiber back, it's pulling the cell forward. And that's how translocation happens. Once you've made this new attachment point, the cell pulls itself in that direction. And then the retracting tail should let go. And these attachment points are focal adhesions, which are really important structures. This is the picture from your book. Focal adhesions are made of integrins and then other proteins. So integrins are the transmembrane proteins. Outside, you've got binding sites for fibronectin, which is extracellular matrix. And then inside, you've got a connection that is going to bind talin and other proteins to connect to the cytoskeleton. And you can see that here. Here's extracellular attaching to fibronectin. Then you've got talin and vinculin, which then attaches to your microfilament. And this is what that myosin is going to be pulling on to pull the cell forward. Here's another important protein here, alpha-actinin. A lot of times when you've got arrays of microfilaments, they're going to be cross-linked, and alpha-actinin does a good job of cross-linking them. We'll see this again when we get to skeletal muscle. It's interesting to me when you see a protein being used for one specific structure, and then you find it's in a totally different structure, but because it's performing the same role. So alpha-actinin is important for cross-linking microfilaments. And here, again, this isn't that important for this exam. I'm not going to ask you about this, but just because in the future we will talk about it. These focal adhesions, in addition to connecting the cell to extracellular matrix, they're also involved in signal transduction. So when the cell is connected to something else, it knows that it's connected, so a cell that's not connected to anything will typically go into apoptosis. So this focal adhesion is important for keeping the cell alive. Okay, so, and make sure you keep in your mind separate cell crawling, which is this guy, from amoeboid movement. Totally different, right? We're not pulling the whole cell. What we're doing is sending out a foot, and if that looks like a good direction to go, the cell just pours itself into that foot, and the foot expands, and the cell keeps moving in that direction. And that happens just by polymerizing and depolymerizing microfilaments. So your microfilaments are being polymerized out in front and depolymerized behind. So, basically you get actin monomers here, and there's like a contraction mechanism with the myosin here that kind of squishes the cell and forces those monomers forward, and then they polymerize out. So forcing it forward is pushing the plasma membrane out, making that pseudopod, and then polymerizing is like solidifying the pseudopod. And it can depolymerize and switch back the other way. If you watch some videos of amoeboid movement, it changes a lot depending on where the cell is being attracted. Okay, so that's it for the review portion of our program today. 11:21. Okay. So what I want to talk about today is mainly skeletal muscle and cardiac muscle contraction. Hopefully we'll get to smooth as well. And so we've been talking about myosin a bit, but we haven't really focused on it much at this point. There are many different myosins. I think there's like 12 or 20 myosin genes, and they're used in different places. A minute ago when we were talking about Filopodia and little fingers, microvilli. We talked about myosin connecting that structure to the plasma membrane. That was myosin 1 here. A very short tail, but an active binding region there, and that myosin is being used structurally. They're not really being used for movement. But some of your other myosins, though, have got a long tail, and they are going to be used for movement. In skeletal and cardiac, this myosin 2 is the one that's being used here. So we've got several parts of this structure that are important. First of all, you've got your head region, the globular head. By the way, can you guys see my cursor moving around? Yes? Okay. So you got your globular head, and that's what binds the actin and binds ATP. That's kind of the business end there. And then you've got a hinge region where it flexes based on whether it's phosphorylated or not. And then you've got the tail, which is structural, and you'll bind to other myosins with that. So typical of so many proteins we see in cells, it's a homodimer, and the tails are wound around each other. So you've got this coiled section that gives it structural integrity there. Okay, the other thing that's important here with your myosins are these light chains. Okay, they're separate polypeptides. They attach to the heads here, and they are going to regulate the ATPase activity of the myosin. And that'll be especially important in smooth muscle, in that the myosin will be just all curled up until those myosin light chains are activated, and then it straightens out. So we'll see that later today, hopefully. So this myosin then, the long tails here will attach to each other. And here you've got anti-parallel arrays, but also parallel arrays here. So they attach to each other, and when they do, it's just spontaneous, basically, because there's non-polar amino acids sticking out here that want to get away from the water solution. So they attach to each other and form these thick filaments. What's important about this is you've got these myosin heads sticking out, and they kind of spiral around the thick filament. So when you look at, if you do a cross section through here, you can see that these are the tails and these are the heads sticking out. So in 360 degrees, there's heads sticking out everywhere. And that's important. If you look over this side, this is an electron micrographic picture of it. Your thick filaments in the center, and it interacts with 6 different thin filaments. Okay, so each thick filament interacts with 6 thin filaments. Each thin filament interacts with 2 thick filaments. You can see that there. So this spiraling around makes it so that we'll see, as the myosin heads start to do cross-bridge cycling to move, there are always heads attached somewhere between the thick and thin filament. So it's not like all the ores get lifted up out of the water and you could lose tension there. There's always heads attached during the contraction. So control of contraction is all about calcium, of course, with skeletal muscle and cardiac and smooth. It's always about calcium. So here is your thin filament structure. So the plus end of the thin filament is always going to be capped with cap Z, right? Stopping it from depolymerizing. The other end, the minus end, is capped with tropomodulin. Again, you need to cap them both because after they've been polymerized, they will have their ATP will be hydrolyzed to ADP, and they're going to be unstable. So those are going to stabilize that thin filament. In addition to that, we've got this long black rope here, which is tropomyosin. Tropomyosin is blocking the myosin binding sites on actin, so myosin can't interact with it. And then you've got the troponin complex, which is controlling the position of that tropomyosin. So when there's no calcium, myosin bind. Well, we'll see that in the next slide. I don't have to do that here. Okay, so that's your thin filament. Structurally, again, once you put this into a sarcomere, you've got a bunch of proteins in the middle that are holding on to the plus ends of the microfilaments here, the thin filaments. So here's your cap Z's embedded in the Z line. This Z line includes alpha actinin. Remember, I told you to come up again. So here's alpha actinin again, attaching 2 microfilaments, holding them together. In the center here, you've also got some intermediary filaments that we'll talk about next, and they're holding that all together. So cap Z, alpha actinin is all in the Z line. So here's our tropomodulin story. So you've got 3 proteins in tropomodulin, troponin T, C, and I. And these are going to control the position of that tropomyosin. So when tropomyosin is in this position, myosin can't bind to it because it's being blocked. When calcium binds to troponin C, that moves the troponin complex, which then pulls the tropomyosin complex, and now myosin can interact. So calcium binding to the troponin C is the trigger that allows skeletal muscle to contract. Now, backing up, we're going to look at here a little bit the ultrastructural aspects of the muscle. I always like to compare it to a rope. If you think about a rope, it's basically you take these really thin threads and you wrap them around together, you get a cord, you wrap a bunch of cords together and you get a thicker cord, you wrap the thick cords together, you get a rope, right? Muscle is kind of the same thing in that a muscle itself is a whole bunch of muscle fibers. Every muscle fiber is a single cell, multinucleated cell, but one cell from one end to the other end of that muscle. Um, and then within a single muscle fiber, you've got multiple myofibrils, which are basically sarcomeres lined up in a row here. So what do we call that? I forget. Anyway, so that's the structure. That gives it a lot of strength. Now, when it comes to, and we'll talk about innervation later, but the functional unit then of this muscle, the actual contractile unit, is the sarcomere. So here's your sarcomere. And this is another kind of historic thing that I think is a little antiquated, but it's still the way people look at it. Imagine this, you're looking through a microscope, it's 1920, you're looking through the microscope at a skeletal muscle, you're trying to figure out how it works. You don't even know what proteins are there, really. So what do? You see bands. So everything gets the nomenclature all has to do with the bands you can see through a light microscope. A striated muscle, right? It's banded. It's pretty clear. So basically, your bands are you've got a Z line. The I band then is this area here that's thin filaments that are not overlapped by thick filaments. Your A band is basically the thick filament. So the whole thick filament is your A band. In the center, you've got the H band, which is thick filament that's not overlapped with thin filaments. And then M line is the line in the middle. So you can see it down here. Your thin filaments only in the I band, overlap here between this in this A band, but here in the middle, the H band, there's no overlap. It's just thick filament. There, just thick filament. Now, M line, you've got another protein that's holding the thick filaments to each other. So that's why you've got extra proteins here. And we'll see that in a sec, so don't worry about it. Okay, so that's your, oh, okay, so here are the other structural components of that sarcomere. So this is the picture from your book, which leaves out some things. I like this better. It comes from my other book, A World of the Cell. So anyway, here's your thick filament, and you've got your myosin cross bridges here sticking out. You've got myomasin in the center that's holding thick filaments together. And that's what I said here in that M line, you've got extra proteins. That's your myomasin that's holding the thick filaments together. You've got your tropomodulin and your CAP-Z stabilizing that microfilament. These yellow lines are the tropomyosin. Now, the other two proteins that are really important for structure are nebulin. Actually, that's the yellow thing, nebulin. Nebulin's wrapping around the microfilament, keeping it straight, right? If that thing curls up, it's not going to work anymore. So nebulin helps to keep it straight. And titin's doing the same thing here with the thick filament. So nebulin and titin are important because they're keeping the thin and thick filaments aligned with each other so they can contract well. Anything else? I guess that's about it. So in terms of contraction then, your I band, anywhere that isn't overlapped is going to shrink. So your I band shrinks and your H band shrinks and go from relaxed to contracted. Okay, so you're losing that. And then here, this is one of my animations. I actually made this animation for oh, that's not it. Nope, that's not what I want. Sorry, just take me a sec here. Okay. There it is. Okay, so here, let's make this bigger. So here's what happens when you activate this. You basically your myosin heads grab onto the thin filaments and pull them closer, and you can see the bands changing in length. So your H zone gets smaller, your I band gets smaller. Let's do that again. That's your sliding filament. Okay, so we're back to this. Wait. Oh, that's chapter 2. I don't need that. Sorry. So that's the sliding filament muscle contraction. So here's what's going on with those little heads. And this is kind of important. I really like the way this whole process is understood at the really minute level. What actually is going on with those myosin heads? Like in general, we say myosin's hydrolyzing ATP, and that's causing contraction. But when you look at it really close, I think the mechanism is really beautiful. So it's a cycle, so you can start anywhere. Let's start here. So this is the Riger complex right here, where the myosin head is bound to the actin microfilament, and it won't let go. So if there's no ATP, the myosin stays attached to the actin there. Now, when ATP is around, it allows it to let go of the actin. It'll then autophosphorylate. So it puts that phosphate on itself. So now it's got both ADP and phosphate on it. And when it does that, it absorbs, basically absorbing the energy from that third phosphate of the ATP. And the myosin head is going to go from like a 90-degree angle up to a 45-degree angle or something like that, 45 up to 90. So that's going from the low energy myosin state to the high energy myosin state. Now, the next thing that happens is the phosphate comes off. When the phosphate comes off, that allows the myosin head to grab onto the actin. And then when the ADP comes off, it goes back to its low energy state. And that's the power stroke right there. So again, it's like ATP picks up the myosin head picks up ATP, lets go of the thin filament, and then autophosphorylates itself. It goes into this high energy conformation. When the phosphate comes off, it grabs onto the thin filament. When the ADP comes off, it then goes back to its low energy conformation. And in doing so, it pulls the thin filament and the muscle shortens. And then again, it's going to be stuck in this low energy state until ATP attaches to it again. And that's your Riger state. And that's why, when somebody dies and their ATP stores and their muscles run down, they get stiff. And then over time, that stiffness will go away as the myosin heads start to break off. But that's why uh, dead bodies are stiff. Rigor mortis. Rigor mortis, I'll say. Okay, so that's what happens when calcium's around. So the next question is how is the calcium controlled? Because that's how you control the contraction, is by controlling calcium. So in a skeletal muscle, you've got a very elaborately made endoplasmic reticulum. So this is your smooth ER basically, but it is structurally different from smooth ER in other cells because its function is so important here. So here we call it the sarcoplasmic reticulum, SR. So this is your SR, and the SR is in what we call a triad junction here. So you've got one SR on one side, another SR on another side, and this transverse tubule, which is an extension of the plasma membrane, comes right down into the cell and attaches to these sarcoplasmic reticuli. And this sarcoplasmic reticulum, of course, is a huge store of calcium. You can see you've got this end of it, which is the terminal cisternae, and then you've got the lateral portion of it. The lateral portion of it has got a lot of calcium ATPase to suck calcium back up for relaxation. And then it stores it and it gets released from the terminal cisternae over here. So, and this whole structure, two terminal cisternae and the plasma membrane here, T-tubule, are right over the area of overlap in the sarcomere. So as soon as calcium comes out, boom, that sarcomere starts contracting. It's a really lovely mechanism. So how are we controlling this now? So it's all about the attachment between this T-tubule and the terminal cisternae of the SR. So you've got two proteins here that are key. In the T-tubule, you've got a voltage-dependent calcium channel, and in the T-tubule, you've got the ryanodine receptor. So the voltage-dependent calcium channel here, gated by voltage, right, doesn't really act like a channel. You don't get a lot of calcium through it. It's mainly just acting as a voltage sensor. So remember when we talked about calcium channels in chapter 2, they've got that S4 segment that's got positive charges on it. So when the voltage on the membrane changes, it moves and it changes the structure of the protein, the conformation of the protein. So here at this T-tubule junction here, you've got the SR ryanodine receptor, which is a huge calcium channel. It's basically a 4-barrel calcium channel. It's sitting right there and it's being plugged by the calcium channel, physically plugged. It's stuck there so that nothing can get out. Then when the voltage changes, so you've got an action potential comes down the T-tubule from the plasma membrane, the voltage-dependent calcium channel changes conformation, and that allows the ryanodine receptor to flood calcium out. And then that goes right to the contractile apparatus and you get contraction. And then when the voltage changes back, that calcium channel goes back in, plugs the ryanodine receptor up again, and calcium ATPase sucks the calcium back into the SR, and you get relaxation. So that's excitation contraction coupling in the skeletal muscle. Really important. It's different from cardiac, and you should know the difference. So here we've got a physical interaction between the calcium channel and the ryanodine receptor. And then this is, I really don't like the figures from your book. This is the, from chapter 2, this is neuromuscular junction in your book. Yucky, yucky. This is a better one here. This is a little video. Let me see if I can share this. This is a nice video. It explains a lot of good stuff about skeletal muscle contraction. Oops. Stop share, screen share.
A skeletal muscle
All right, can you see this animation with the skeletal muscle? Yes, okay.
contracts only when stimulated by nerve impulses from a motor neuron. Without innervation, muscles become paralyzed and eventually atrophy. The axon of a motor neuron usually gives out many branches, supplying multiple muscle fibers.
Kind of important to note here, this one muscle neuron is doing a bunch of muscle cells here, bunch of muscle fibers, but not all of them. Okay, you've got many different muscle neurons, and they're interacting with different cells here. Each one of those is a motor unit. She'll talk about that now.
These fibers contract in unison when activated and constitute a motor unit. A motor unit can contain anywhere from just a few muscle fibers to thousands of them. Small motor units are found in muscles that require finer control. For example, muscles that are responsible for subtle movements of the eyes. Large motor units are found in larger muscles that require strength, such as muscles of the arms and legs. Muscle fibers of the same motor unit are usually not clustered together, but scattered throughout the muscle. This way, stimulation of a motor unit has a slight but uniform effect on the whole muscle and not causing a small localized twitch.
And this, well, I'll talk about this again when we get to skeletal or cardiac. The way you get a stronger contraction in skeletal muscle is you activate more motor units, right? So every time you stimulate a skeletal muscle cell, it contracts as hard as it can. And you get a softer or stronger contraction based on how many motor units you recruit. Cardiac's different. And we'll talk about that when we get there.
The strength of a muscle contraction is determined by the number of motor units that are activated
said
at one
that.
time. Even at rest, most muscles are in a partial contraction state called muscle tonus, which is maintained by alternating activation of a small number of motor units. The connection between a motor neuron and a muscle fiber is called a neuromuscular junction, which is basically a chemical synapse between the nerve terminal and a specialized area of muscle cell membrane called the motor end plate. When an action potential reaches the nerve terminal, it causes the release of the neurotransmitter acetylcholine into the synaptic space. Acetylcholine then binds to nicotinic receptors on the end plate. Nicotinic receptors are ligand-gated ion channels. Upon binding to acetylcholine, they open to allow sodium to enter the cells, depolarizing the cell membrane, producing the so-called end plate potential. An action potential is generated in the muscle cell only when the end plate potential reaches the threshold required to activate voltage-gated sodium channels located outside the end plate in the neighboring membrane. When activated, these channels allow faster influx of sodium, further depolarizing and eventually reversing the polarity of the cell membrane. At this point, voltage-gated potassium channels open for potassium to move out, quickly returning membrane voltage to its original resting value. Once generated, the action potential spreads like a wave, thanks to similar voltage-gated ion channels located throughout the muscle fiber. The action potential also runs deep into the fiber via T tubules to reach the sarcoplasmic reticulum. Here it activates voltage-gated calcium channels, releasing calcium from the sarcoplasmic reticulum into the cytosol of muscle cells. Calcium then sets off muscle contraction by the sliding filament mechanism. This mechanism is described in another video. Another important component of the neuromuscular junction is the enzyme acetylcholinesterase. This enzyme removes all acetylcholine molecules that do not immediately bind with the receptor and those that are done activating a receptor. The enzyme action essentially terminates synaptic activation, giving the muscle time to relax and thus preventing continuous contraction that would result in muscle spasms. Substances that cause muscle weakness or paralysis do so by interfering with the function of neuromuscular junction. Botulinum toxin prevents acetylcholine release from the presynaptic side of the junction. Some other toxins attach to nicotinic receptors, blocking acetylcholine from binding but do not open the ion channel. Certain drugs lodge into the channel of nicotinic receptor, blocking the passage of sodium. All these substances prevent activation of muscle cells and cause flaccid paralysis. On the other hand, some pesticides inhibit acetylcholinesterase, preventing degradation of acetylcholine, causing continuous activation of muscles. That's how they induce muscle spasms and cause spastic paralysis.
Okay. So let's go back to my PowerPoint. Okay, so that's your motor end plate. And let's see, next slide. Basically, there's your motor end plate. You've got nicotinic receptors here. When this motor neuron releases acetylcholine that activates them, nicotinic receptors are ligand-gated channels. They pass sodium mainly. So sodium comes in, causing depolarization. So that end plate potential is similar to an EPSP that we talked about in neurons. So one small depolarization. If you get multiple of those, then your voltage-dependent channels that are nearby get activated. Then you get an action potential. Now with skeletal muscle, that'll go across the plasma membrane, but it'll also come down through this T-tubule, and that's where you get that interaction of the Voltschmidt calcium channel and the ryanodine receptor that allow calcium to release. Calcium then binds to the troponin complex, moving tropomyosin, and then you get cross-bridge cycling with the myosin until the action potential goes away, and then the calcium is taken back up into the SR. Something I forgot to say, I just want to back up real quick. Inside that SR, you have some calcium binding proteins. You can imagine if you put a whole lot of positively charged particles in one place, it's not going to be very happy. So this calsequestrin then helps to neutralize some of that positive charge, so you can get a lot of calcium into the SR. Okay, so that's skeletal muscle contraction, skeletal muscle EC coupling, which is just here in the triad junction. Now, cardiac, as I alluded to before, is going to contract differently than skeletal. And so the mechanism of EC coupling is going to be different. Big difference here with cardiac muscle is every time the heart contracts, every cell has to contract. And that keeps it coordinated. So it has a coordinated action. So with the skeletal muscle, when you want a stronger contraction, you activate more cells. In the cardiac, when you want a stronger contraction, you make each cell contract harder. We're not going to talk about that here, but when you get into cardiovascular pharmacology, they'll talk about that a lot, how you stimulate each cell to contract harder, and that's inotropy, basically. But anyway, where the cardiac muscle contracts is you've got a spontaneous depolarization here in the right atrium, the SA node. And if you take a heart out of a body, it'll keep contracting because it's self-sufficient in terms of its stimulation. You know, skeletal muscle won't contract out of a body because there's no nerves attaching it. But here, you know, I've had a human heart in my hand beating before. I was in, but it's okay. It was legal. I was in on a heart transplant in Cincinnati, and we got the old heart when they put the new heart into this person. So there I am with a human heart beating in my hand. That was probably one of the coolest things I've ever done. But anyway, your stimulation starts here at the SA node, and then it travels from cell to cell across the atria, and then the atria contract. But then there's this insulation basically between the atrium and the ventricle. So the atria contract, and then that depolarization hits the AV node, which then depolarizes. And this is more like conducting than contracting tissue. So that takes this action potential down to the bottom of the cell through these bundle of His here, and then comes back up and then spreads into the ventricle. And then it starts going cell to cell again through the muscle of the ventricle. So that's kind of important. I guess it's not important here. This is cell biology. This is physics. Whereas what I'm talking about is physiology. But basically, you're bringing that action potential down to the bottom of the cell, and then it's coming back up and going cell to cell from there up. And that way the heart contracts from the bottom up. So squirting the blood out and up. Well, not out in general, because then that you would die, out into the aorta. Okay, and what makes that possible is the connections here between these cardiac cells. Cardiac cells are kind of smaller. They're smaller than skeletal muscle cells, and they're kind of boxy, and they're attached to each other by these intercalated discs, which are really strong connections between the cells, because they're gonna undergo a lot of mechanical stress. So they're attached to each other by these connections. Desmosomes are also connections between cells. So the desmosomes are more structural connection, and then your intercalated discs have got these gap junctions that are part of them. And the gap junctions then are basically ion channels between cells. And that's what allows the action potential to go from one cell to another and spread over the atria, and then spread down the connecting tissue, and then across the ventricle. It's just the action potential just going cell to cell through these gap junctions. What else do I want to say here? I guess that's about it. So basically, again, when the cell contracts, when the heart contracts, every cell contracts. And so, because we've got this need to have every cell contract, this EC coupling is going to be different here in cardiac and skeletal muscle. So here in cardiac, the calcium channel is actually acting as a channel, and it's comes from a different gene than the skeletal muscle calcium channel. So it's a totally different protein, and it does act as a channel. So when the action potential comes down the T-tubules, there are T-tubules in cardiac as well, the channel opens, and that allows calcium to come in. And that calcium is called trigger calcium. Okay, it's not enough to contract the cell, but it's enough to come in and open the ryanodine receptor. And the ryanodine receptor then causes calcium to flood out into the cell, into the contractile apparatus, and you get contraction. So that's excitation contraction coupling in the cardiac. The action potential comes down, opens that Volchmann calcium channel, and calcium comes in and activates the ryanodine receptor. You get the flood of calcium. That's called activator calcium. So you've got trigger calcium and activator calcium. The activator causes contraction of the cell. And then you've got Circa over here pumping calcium back into the SR. And then you also have sodium calcium exchanger at the surface taking calcium out. So I talked about this last time a little bit, but I'm going to throw it out here again. Your NCX is kind of a low capacity, fast exchanger. So it brings calcium down quickly, but it doesn't have a lot of capacity to it. Your Circa's high capacity, but works slower. So that's why you have two different mechanisms here. And then later, I don't know, I don't think we're going to talk about this in this class, but like I said, when you get into cardiovascular pharmacology, you'll see when you want a stronger contraction, basically you're going to phosphorylate this calcium channel so that more calcium comes through on each action potential. So you get more trigger calcium, which causes more activator calcium to come out. So you get a stronger contraction out of it. And you also phosphorylate phospholamban, which makes the Circa pump faster. So you get a stronger contraction and a faster relaxation. I don't know. That's not important for this class. Hopefully you'll get that in cardiovascular form. Okay. What is important? Okay. So that's it for cardiac and skeletal. So skeletal, excitation contraction coupling, the Volchmann calcium channel is acting like a plug in the ryanodine receptor. It's physically opening that channel. In cardiac, the Volchmann calcium channel is acting like a channel. Calcium's coming in. You get trigger calcium. Trigger calcium then activates the release of activator calcium, and that's what causes contraction. Now, smooth muscle. Let's take a breath here, because smooth muscle is very different. This is going to be a very different conversation here. So smooth muscle, the nice thing about smooth muscle is it takes very little energy to contract it. Skeletal muscle and cardiac use a lot of ATP to contract. Smooth muscle, much less, but it's going to contract a lot slower. It's skeletal muscle can contract and relax in 1/10 of a second, but a smooth muscle might take 30 seconds to fully contract. So it's a lot slower than skeletal and cardiac, but it can hold tension without using too much energy. If you pick up something heavy with your skeletal muscle, they're going to get tired and you're going to have to drop it eventually. Smooth muscle will hold that tension for as long as it needs to. And smooth muscle cells are kind of spindle shaped, and they kind of overlap each other, and they go kind of around the organ that they're surrounding. So smooth muscle, you're going to find in arteries and veins. So they're going kind of circularly around that artery and vein. They're going to control the diameter of it. They are also found kind of around the bladder. So when they contract, the bladder contracts and helps you to expel urine. So they're kind of, I guess you would call that transverse across the lumen of the vessel. But anyway, they're overlapping like this, and this is relaxed and this is contracted. You can see it looks like there's ropes tying it down. It's just like, for Christmas you get you get a rib roast, a boneless rib roast, and and the butcher ties it up. That's kind of what it looks like to me. It's like being tied up by these ropes all the way across it. But what those ropes are is these structures here. So you've got little dense bodies connected by intermediary filaments, desmin and vimentin, and also you've got myosin in the center, thick filaments here. So it's all kind of on the membrane there. With these dense bodies are on the plasma membrane. And then you've got a mechanical junction between cells to help with the mechanical stress that happens when it contracts. So that's kind of the physical structure of it. Now, here's how it's controlled is interesting too. So with skeletal and cardiac, we're just controlling the thin filaments, right? With the troponin complex there. Here, both the thick filaments and the thin filaments are going to be controlled. Your thick filaments are controlled by phosphorylation of those myosin light chains. In smooth, then, when the smooth muscle is relaxed, the myosin is kind of all curled up. The tails are not straightened out. When the tails are straightened out, these myosins can overlap each other on their tails and form fibers, but when they're folded up, they can't. So when the cell is stimulated to contract, calcium goes up in it again. It's calcium again, and that activates a myosin light chain kinase, an enzyme that will phosphorylate those light chains, and that causes it to straighten out. Once it straightens out, it forms thick filaments, and then you can get contraction. The actin is also going to be controlled. We'll see that in the next slide. Okay, so the first part of this is what we're going to talk about in the next chapter, so it's not that important here. I guess I talked about this today already, phospholipase C. But typically, a smooth muscle, like let's say an arterial smooth muscle, is going to be controlled hormonally, whether it's your sympathetic nervous system releasing norepinephrine, or local modulators like prostaglandins or something can be released, and they'll activate cell surface receptors that will then turn on this phospholipase C mechanism. So your receptor binds the hormone. And again, don't worry about this part. The next part's what matters for this chapter. But receptor binds to the hormone, that activates the G protein. The G protein activates phospholipase C, which then breaks down PIP into IP3. Okay, from here, we're going to pick it up. Here's what's important for this test. So IP3 gets released based on this phospholipase C activity. IP3 will then come to the IP3 receptor. And again, reviewing from chapter 2, we've got two calcium release channels on the SR. One is the ryanodine receptor, which is calcium activated, and the other is the IP3 receptor, which is activated by IP3. So IP3 gets released, binds to the IP3 receptor, which is a channel, and calcium comes out. So now calcium's gone up in the cell, and it's going to bind this guy, calmodulin. Okay, so we've seen calmodulin before when we were talking about the structure of Philopodia and the structure of microvilli. Here it's not being used structurally. Here it's part of a signal transduction chain. So calcium comes out, it binds to calmodulin. Calmodulin then goes off to activate other things. So calmodulin will bind to myosin light chain kinase and activate it. Myosin light chain kinase with calmodulin then will phosphorylate the light chains, and then the myosins straighten out, they overlap, and now you've got thick filaments, and this is ready to go. On the other end, here's your thin filament over here. You've got another protein here called caldesmin. So caldesmin is kind of playing the role of tropomyosin, role of tropomyosin in skeletal and cardiac. It's blocking the myosin binding sites on the thin filaments. And there's two ways to get that off. One is by phosphorylation by another kinase, protein kinase C, that will pull it off. But also calcium calmodulin will bind to caldesmin and pull it off. So This is all calcium dependent again. Protein kinase C needs calcium to be active. So again, calcium increases, that causes caldesmin to come off by either phosphorylation or calmodulin. And now you can get cross-bridge cycling and contraction. So you can see why this takes so much longer to contract. With skeletal and cardiac, you open those ryanodine receptors, calcium goes right on the contractile apparatus, which is all ready to go, and it contracts. Here, you've got a whole bunch of enzymatic reactions that have to happen. Remember, phosphorylation takes a little bit of time, right? So this is a much slower mechanism than the other. But once it contracts, then it can hold on almost like a rigor state without using any ATP. So it can hold a lot of tension without having to burn ATP. So let me go back. Let me just go over this again with talking less and making it go faster. So your hormone then hits the receptor, activates the G protein, which activates phospholipase C. PLC breaks down PIP into IP3 and diacylglycerol. Diacylglycerol will activate protein kinase C. IP3 will activate the IP3 receptor. Calcium comes out, calmodulin binds, myosin light chain kinase gets activated, myosin light chains get phosphorylated, and the myosin is activated. And on the other side, diacylglycerol activates protein kinase C, which phosphorylates caldesmin, releases the thin filaments, et voila. You get contraction. Okay, so that's smooth muscle contraction. What the hell time is it now? 12:04. Guys have any questions about this? All this muscle contraction stuff? Are we all good with this? Yes? Okay, in that case, one more thing I want to talk about today before we go, before we finish for today. And that is your book talks about the spectrin membrane skeleton. So basically, this is the structure that's like right under the plasma membrane that's holding the cytoskeleton to the membrane. So I talked about right underneath the plasma membrane is the cell cortex, which is a 3D mesh of actin. This is attaching to the plasma membrane by this spectrin membrane skeleton. And this is the basic structure down here. This is, again, from that other textbook. It's majorly simplified. This is a little more complicated way of looking at it. But the proteins that are important here include this guy in the middle, band 3, which is a chloride bicarbonate transporter. Band 3 will bind ankyrin, and chirin will bind two different spectrin molecules. So these long rope-like things are spectrin. Spectrin is a dimer of alpha and beta monomers, and they are attached here to ankyrin, and ankyrin's attached to the band 3. And over at the other end of the spectrin and chirin, you've got band 4.1, which is helping to attach the spectrin to this protein, which is glycophorin. Glycophorin. I love sugar. It's my daughter. She loves sugar. But it's also this protein. And it's called glycophorin because it's got a lot of glycosylations on it. Remember, we talked about glycosylations being important for immune recognition of cells. So glycophorin then binds to this band 4.1 and aducin, and an actin protofilament, and that's all part of this structure. So you've got this basic structure here, band 3 and chirin spectrin going over to band 4 glycophorin and an actin microfilament here. And that basic structure repeats over and over again and it just kind of repeats itself in 2 dimensions across the membrane. And so what this is doing, back up for a second again, you've got transmembrane proteins that are going to keep this structure anchored to the membrane, right? It's not going to come into the cell. It's anchored to the membrane by these transmembrane proteins. And then you've got all this stuff that can then attach to the actin cortex. This, I don't know, I just don't like the figures in your book. I have a hard time seeing anything here. But this I think makes it a little more clear. And let's do this. Let's finish by watching a little bit of this. This is a really great animation. It's old too. I think this has got to be like 12, 15 years old, but I've never seen a better view of what it's like to go into a cell. So let's take a look at this. Can you guys see this inner life of the cell? You do see it. But do you see the animation or just the figure?
It's not playing. I can
Oh.
just see the screen.
Okay, let me change the share then. Okay. So it starts out, well, let's just skip over this because it's stuff that we'll talk about someday, but we're not ready for that yet. There's where we want to start. So now the camera or your mind's eye basically is going into the cell. And when you do there, here's the structure we're talking about. This is spectrin, and this is ankyrin attaching to band 3, and in here is glycophorin and adenosine, and all those proteins are there. And that's underneath the plasma membrane. And now here, this is what I keep talking about with gel actin in the cortex. Watch this 3D mesh as you go through. So you can see how that would restrict movement of things and also support the plasma membrane. Now this is polymerization of microfilaments. You can see filament cross-linking them there. That yellow is filament. Here comes gel solid cutting a microfilament in the middle. Now this is microtubules, which we'll talk about in the next chapter, in the next lecture. And this is a molecular motor pulling a vesicle along a microtubule. So we'll talk about this in the next class. This is your centrosome, and microtubules polymerize off that centrosome and go all over the cell. And then, these vesicles can pull things along those microtubules to travel things on. We'll talk about that in the next class. It's not that important. 12:10. Okay, I think that's about all I want to talk about today. Are there any questions or comments? No questions? Okay, well, I will, I'll put that, put the recording up in Brightspace today. You can watch me all night tonight. I'm sure that would be fun. And I'm gonna figure this damn Microsoft Teams out again. I don't know why I wasn't allowed to share my screen, but something else I gotta figure out. Okay, so that's all I got for you guys today. Goodbye, everybody.